Populations EBV session called From Data to Decisions, Advancing Biodiversity Monitoring through Species Populations EBVs. I'd like to start by introducing the organizers of this session, and starting with Elkin Norio, who will be helping us through the session, and he'll be introducing the talks and has done a lot of heavy lifting for the session, so for organizing, so thank you very much, Elkin. I'd like to say something, you would like to introduce yourself, okay? Walter Yates, if you could wave your hands, everybody knows who you are. Elena Alea. Nereid Euro. Thank you. Laura Garcia. Juan Carlos Rey. Okay. Well, great team of organizers, so thank you very much. Right. I'm going to give a very exceedingly brief introduction to the session. Get on to the first slide. I'm not going to talk about what is a species population EBV. Walter Yates will cover that in some detail in the second talk. But I would like to start by just, many scientists have joined this journey of developing and implementing species population EBVs over the course of time, and I think it's helpful so that we're all on the same page to understand a little bit about where it came from. So I'll very briefly give a bit of a landmarks along the way in the development of species population EBVs. So this all started going in 2013, and actually slightly before 2013, but the first publication that came out was this paper by Pereira et al. Called Essential Biodiversity Variables. And this was produced by a group of geomon researchers that had got together, and over some time this idea percolated, and this publication introduces the concept of essential biodiversity variables and provides the reasoning for why they're necessary and really critical if we're going to upscale biodiversity modeling and deliver it effectively and in closer to real time to decision makers. And so this is essentially in the literature where the idea of essential biodiversity variables started. And then a few years later, there were two papers. This paper by Daniel Kisling and colleagues started to have a look at, well, if we're going to do this, how are we going to actually operationalize it? What sorts of things do we have to start thinking about to operationalize, specifically now, species population EBVs? So species population EBVs are one out of 6 classes of essential biodiversity variables, including genetic, traits, communities, ecosystem, structure, and function. Thank you, Walter. And then in 2019, there was this paper by Walter and colleagues that essentially put out a vision for where we were going with species population EBVs and what the realization at scale of species population EBVs would look like. It provides the definitions for the two EBVs underpinning species population EBVs, species distribution and species abundance, and a roadmap, if you like, for species population. So, and now 5 years later, or 6 years later or so, there's an EBV data portal, for example, with some realized examples of species population EBVs. There's over 300 primary publications related to species population EBVs, and some of the sorts of themes, if you like, are looking at how we implement EBVs at different spatial scales. There's some thematic focused areas, for example, taxonomic, birds, but also invasive alien species, and how that is supported by the species population EBVs. There's a lot of work on automatic workflows to produce them, species population EBV data cube, and also then making these products available for policy and conservation decision making. And I think today we're going to hear some of the most recent and exciting developments about and related to species population EBVs, and I think it's going to be really valuable to think about where to next, and hopefully we'll have a bit of a discussion at the end of the session for the panel about that topic. So I'm now going to hand over to Elkin to take it further. Thank you so much.
So our first speaker is Tom Howard.
Thank you. My name is Tom Hauer. I am a science product manager in the Center for Avian Population Studies at Cornell Lab of Ornithology. I'm going to talk today about how we generate species population EBVs by standardizing participatory science data through machine learning workflows. Also track avian population dynamics. I want to acknowledge all the co-authors here. There we go. So this participatory science product, as many of you all probably are aware of, is eBird. We have over 2.1 billion bird observations now, covering large parts of the globe, although there are obvious spots in this map that don't have coverage. But eBird is essentially a participatory science project that works to collaborate with networks of birding enthusiasts to collect data about birds so that we can understand distributions, abundances, and trends of birds. But the tricky part about this is the fact that these are humans collecting this data. These are not machine sensors. And so there's a lot of variation and noise and bias associated with how this data is collected. And that presents a number of challenges. So I'm going to talk to you a little bit about how we address those challenges, what the products we generate, and how they relate to EBVs are, some applications, and then wrap up a little bit of future work. Does it work? Oh, it works. So you prove it over the last session. So the challenges, fundamentally in the top right, we want to understand the spatio-temporal associations between birds and their habitats. It's a like fundamental ecological challenge, understanding those relationships. But along the way, we have a number of challenges associated with that human observation process that we need to deal with. And the overall framework for doing this is a species distribution model, lots of species distribution modeling here. We want to estimate information about birds. And so we need to use that primary bird data. And this is where the eBird data comes in. And we do have a robust data quality process behind all of the eBird data that uses humans to evaluate records that might not be plausible. We then filter that data down further to data that is suitable for scientific use. For example, if somebody was traveling a long distance or was out for a long period of time, it doesn't fit well into machine learning workflows. And we're using presence-absence data. eBird checklists are considered complete, so that if somebody goes out, they're telling us whether they reported all the species that they were able to identify. And through that, we can make some inference about the zeros on the checklists. We then, ooh, too fast. What is happening? Okay, we combine that with environmental features, common part of species distribution modeling. There are over 150, 170 very high spatial and temporal resolution features describing things from abiotic like elevation through land cover, land use, as well as human features like roads and nighttime lights. And what this lets us do is that when the models learn these associations between where birds are and those features, we can then use that information to make predictions, interpolations to places where people have not been predicted. Spatial bias is still a problem, as that map previously showed. There is a habitat bias towards urban areas where people can get to, accessibility. One way we have developed to ameliorate this is to do a grid sampling method where we put down 3-kilometer grid and randomly sample checklists from that grid. It does not completely absolve us of our urban bias, but it does ameliorate that effect substantially. And then observer variation. This is essentially the fact that every person that goes out birding does it a little bit differently. It has a different experience level, different skill level, and we can compare the rate at which people accumulate species on a checklist across people in space and time, and calibrate them against each other in terms of their ability to detect species. There's a number of papers led by Ali Johnson and others describing this method and its application, and it really helps us control some of that variation between each other. We then wrap this all together in what we call the spatio-temporal exploratory model. This is a way to deal with the fact that bird habitat associations change through space and time. So we're partitioning the globe into space-time chunks and fitting independent models within those unique predictions. We're doing this in an ensemble method that lets us do it many times to get randomization and spatial symmetry. So this is our general modeling framework for dealing with this participatory science. It does let us generate species distributions. For example, we have almost 3,000 species globally now, and we increase that number every year. Fitting under the species distribution EBV, we have year-round distributions as well as range boundaries, as well as encounter rate estimates, which are not every year. And we have data products that we are going to be updating every other year at the end of the year. Additionally, we generate a weekly estimate of relative abundance. So I'm going to play this animation here for cliff swallow. Dark pixels are high relative abundance, yellow pixels are low relative abundance, and we capture most of the full annual cycle for species throughout the Western Hemisphere in this example. And this is done at a 3-kilometer by 3-kilometer spatial resolution. And these fit within the species abundance population. So what can we do with this? Well, if we take each one of the pixels on that previous map and divide it by the sum of all the pixels in a given week, we can calculate a percent of population at each location. It becomes a really valuable metric in conservation decision making. And in this example, we're working with a conservation collaboration in the Pacific Northwest that is interested in understanding what their priority species are for conservation action. So if we do this across a suite of species, which are the bar charts here on these two plots, we can estimate a stewardship responsibility metric so that these groups can understand what species to focus on. And there's a paper that was just accepted by Laura Farwell and others describing this application. And here we can see that this species, black oyster catcher, which spends 100% of its populations within this focus area in the Pacific Northwest during its post-breeding migration, making it a key priority species for this conservation collaboration to focus on. We can also take that spatio-temporal information and stack it all together. So we've worked in partnership with the U.S. Fish and Wildlife Service, our main federal agency in charge of regulation of species, and identify the most important locations for these different species groups. So this map is showing for 49 different raptors in dark, high importance, in light yellow, low importance. And this sort of high spatio-temporal resolution stacked together gives a really quick picture of where species like these groups are vulnerable to things like infrastructural development, such as power lines or wind energy, which makes it really valuable for regulatory needs. Okay, we also generate population level trends for bird species. We have these for over 850 species now. They're at 27 by 27 kilometer spatial resolution, so a bit coarser. These are depicting an average trend over a 10-year period. And so on the right here, you can see an example where each dot represents one of those pixels. The size of the dot represents the abundance during that period. It's red, it's decreasing. It's blue, it's increasing. And the deeper the color, the stronger that trend. We are also actively working on developing annual indices as well, so that we can understand more fine-scale temporal information about these trends. But trends present an interesting quandary in terms of analyzing participatory science data as the human observation process gets more complicated because it's changing over time as well. So you can think of things here like checklist density for eBird from 2007 through 2018, growing and getting more adoption. spreading out spatially and growing in density. This sort of pattern could cause a confounding signal in the trend and generate spurious trend. You can also think of things like Merlin sound ID, Merlin identification tools or apps as changing both the way that people learn and grow, but also the pool of people who are participating in eBird, moving from not just people who are obsessed with the eBird app to a broader group of people. And so we deal with this through what's called the double machine learning method, described by Daniel Fink and others in a recent paper. This uses a causal machine learning method. It uses the same information that we use in our status production of eBird observations, habitat participants. But the key points here are that it controls for confounding in the observation process. I don't have time to go into the details of that, but it does let us control for that with a propensity score model. And then we can also capture the spatial variation with habitat features. We can't with other types of regional trend. So I just want to talk about an example of how we use that. You're great. Collaborating with the U.S. Fish and Wildlife Service again, they're focused on golden eagle monitoring in the western United States, and interest in the population size. They're looking at these regions across the western U.S. as management regions. They have been running aerial surveys for the fixed-wing aircraft. You can see those locations, the red dots, and then their estimates of density across these very large management regions. And the problem with these fixed-wing surveys is that they're dangerous, they're expensive, they can't always be run. And so we work with them to integrate our eBird trends with the aerial survey data. And so, for example, in this region, the dot and whiskers represent the estimates from aerial surveys only, and green represents integration. And that gives us confidence that when these surveys weren't able to be run or are not able to be run, we can fill in the gaps with data. Okay, I want to just wrap up by talking a little bit about our future work, extending to deep learning distribution modeling. This is similar to our machine learning-based workflows where we're using the same observational environmental data, but now using a deep learning framework. And the real advantage here is that we can also use species interaction information. Models can learn how species are interacting and how that affects distribution and abundance in a way that those other species by species modeling methods can't. And so we can generate from that rich information about community abundance, taxonomic diversity, This framework also lets us take advantage of multimodal data sets in the future, which really open up the window to help fill in places that don't have a lot of data or species that are hard to detect. So we can use vision modeling methods to take information like spectral data from the satellites and hyperspectral satellites, as well as combine with things like camera trap data or passive acoustic monitoring. And do it in a way where we can take advantage of these large data sets in a raw form as opposed to needing as much labeling and looking at those specific realities. And so I'd like to just wrap up by saying that we can generate these species population and community composition EBVs reliably through these science data by using these machine learning workflows to control for that when it works. Thank you.
It's for Kristen.
Can you talk more about the Merlin effect? And I mentioned the version more lines. So it does
Yep. So right now we don't like take Merlin information directly into our modeling workflows. We do track whether Merlin has been used on a neighbor checklist or not. And we're starting to do research on understanding where that effect is strongest. Not surprisingly, there's a big difference between species that vocalize and species that don't vocalize and the effect that has on the detection. We're not directly controlling for that right now, but we're doing research to do.
I think on the box you showed, you had uncertainty gray. Yeah.
Yep.
How are you judging uncertainty? What is the criteria?
Should come to my last stop. We have a kind of a separate modeling process where we take all the data run a separate modeling workflow where we describe both spatial coverage and then the likelihood that a pixel would be have data given its habitat configuration, so by the classification model. And then we threshold those and basically mask out any predictions from those locations. And it lets us control both general spatial extrapolation. You can think of like, Russia versus places that have not had data in terms of their uniqueness in the habitat. So, obviously, that's
Thank you so much.
Our next speaker is Walter Jess. You have one recommendation for question and responses. We need to rotate the microphone so some people can contribute.
Great, thank you. And it's a real privilege to follow up on Tom. We had Tom with us for a year and a half, 2 years.
like that.
Yeah, and it's great to see how far you've come with the awesome work. Training with us or training us in data visualizations back then as well. So real awesome to be here with you all. And this was initially intended to be the first talk, essentially giving a bit of an introduction to population EBVs. So please bear with me and bringing us back a little bit to the basics. Somehow the sequence ended up the way it is. But at least for some of you, it might be helpful to just go a little bit deeper into the conceptual and then the more methodological aspects of pulling EBVs together. I think eBird is a prime example of not just the distribution aspects of the population EBV, but also particularly the abundance aspects of getting at that. There are some other great examples from the marine arena, Martin Pinsky's work and others out there that have been doing great work with fisheries data. But we remain very data limited. But before we go there, let me take us back a bit and think more about why we are caring about EBVs and a particular definition of species population EBV that, as Melody said, got developed some 7 years ago initially through the GeoBon working group that she and I have been in our co-chairing on species population EBVs. I want to approach that today a little bit from a different angle that got informed by a workshop we had 2 years ago at GeoBon. In association with that, it was sponsored by the Business Earth Fund and the Wilson Biodiversity Foundation. And it was a bit of a different setup where we actually brought in a lot of folks from the user, the consumer community. So the folks that actually are interested in products, not just indicator products or metrics, but also products for planning. And let me therefore approach that slightly differently and maybe very basic way, very mundane way, but let's just bear with me here, please. We are here in Madagascar looking at a spiny forest. You all know how restricted some of the biodiversity there is, how threatened it is, but in terms of pure significance, we can first think about, well, what sort of general significance does this place have in terms of ecosystem services, in terms of nature benefits for people, etc., etc. And then there are these usual ones for that apply to forests. We can then separately look at the particular unique aspects that this place has that talk more about the global distinctness of this location, right? The species that don't occur anywhere else, the narrowness of that particular ecosystem type. That's a very, very small ecosystem. And we can really cast these in terms of a local biodiversity significance and a global biodiversity significance. I'm obviously oversimplifying, right? But it's helpful maybe to sometimes remember that distinction, because in some of our discussions here, and certainly in the broader community out there, as we engage with the user community, industry increasingly, but also government agencies, people out there broadly, these things sometimes get mixed up. They talk about biodiversity, and then we are not, I believe, sufficient to distinguishing those two different things that we mean sometimes when we talk about biodiversity significance. Move to Eurasian temperate boreal woodland, and here we have similar sorts of local biodiversity significance, very important services conveyed by that system, also functions, however, a much more reduced global biodiversity significance. The ecosystem extends over much of Europe and Asia. The species occurring there have a very, very large range. There aren't any endemic species there. And by the way, instead of the Madagascar forest, I could have just talked about the mountain tops here, and a lot of endemic species just as you go a few dozen kilometers away. But this is different for many temporary locations. So this is old stuff, of course, you all know that. But putting this together, it's helpful to appreciate that we have this very similar local biodiversity significance, but a very different global biodiversity significance. And as we were to lose one, we lose much less here compared to there. And if we were to lose A, that would be a dramatic reduction, right? If we were to lose A here, the reduction isn't quite as large, because there's a lot of complementarity, redundancy in these services. And when we then talk about the compound biodiversity significance, local plus global combined, it's clear that we would have a much greater loss in the top one than the lower one, A versus B. And why is this important? It's important for two reasons. It's important from a pure calculation perspective, and it's important from a user and consumer perspective. From a calculation perspective, I think this is one of the more compelling reasons, one of the more compelling ways to convey the importance of the species population EBV, because only if you have the true EBV captured are you able to address global biodiversity significance. And as a reminder, EBVs, the species population EBV, is defined as capturing the occurrence or the abundance of each element of a particular taxonomic scope, say all birds or all mammals or this particular family of amphibians, over the full extent of that particular group. So not just in one country or in one location, but really over the full extent. And if it's a globally occurring, near globally occurring group, such as birds, that would be the globe. So you don't want to leave a species behind or an ecosystem type behind as you capture this information, because if you do, then you're not able to represent the global significance of a particular location. So you can't leave an element behind, and you can't leave a global location behind, because you might not be appropriately appreciating how range restricted the species is. Tom talked about stewardship earlier, right? That's the concept that we're talking about. It's the stewardship of a particular location for the species that it holds. You can only appreciate if you have accounted for all the species in a particular group, say all vertebrates, all trees, as well as their full global extent. So if we want to quantify that stewardship, that importance, the global significance of a particular location, we need the species population EBV. For the local significance, you do not need that necessarily. You can measure the biomass of food, the carbon sequestration that it's doing, etc., etc. Right? You can just do a local measure and you get one. So why does this matter? It matters because there's a whole suite of obvious users of that sort of information, and this is what came out of the workshop. We structured it a little bit, and obviously this is all already quite familiar to you, but it's worth defining in a bit more concrete way. There is a whole suite of use cases for assessment and monitoring, and then separate, there's a whole additional set of use cases for decision support. And it's that decision support in particular that requires then not just that measurement at a local scale to be globally informed, but it also needs that local measurement over a large spatial extent, over a whole country or a whole region, in order to be able to do spatial planning. So all of these particular actors require this type of information, and they have respective area scopes, and several of them are global, some of them are just local or regional, but even if they're local or regional, they often have a requirement for not just the local significance information, but also the global significance information. So here, that's the significance scope. So in red, use cases, the portion of use cases that require global information, and then in in blue those that are fine with just local information. So in many cases, businesses or financial institutions, they might only require, in terms of their own dependency on data, they might only require local information. But if they care about their exposure, their risk, they require actually the global relevance, the global information for that particular approach. Do you have threatened species? Do you have a species for which you're the sole steward or ecosystem? By the way, if you're interested in some of these figures, they're now online, and several folks, Maria, Melody, others in the room here, were involved in this workshop at the time, call them out for their contribution. So it's in this context then that maybe we can recast a little bit the reasoning behind why we are doing this modeling. And I think Tom did already a great job in explaining what this is all about, right? This is about capturing many species or ecosystems in their spatial and temporal distribution. And it's from that information then that you can go to a whole range of use cases that are around indicators and metrics of trend, as well as support planning. And we essentially need models because everything in terms of the raw data is sparse and biased. Right? So overall, we need something like this ecosystem here, where we have a whole suite of actors that are sometimes also users over here that are able to bring data into the fold. We need then a whole suite of communities, and this community here is a very central community, right? That thinks about how you bring this data together, but then more than that, to harmonize it and improve the quality. And then we need to go to this metric development and map development that can then support the whole range of use cases. So switching over for the last few minutes to work out of Map of Life. So kind of switch, now talking about some own project activities here. And we'll hear a little more later from Beth in this session here about how we are trying to sort of fill this middle here. Now, many other actors, and eBird is one of them, right? That try and make that connection from raw data to then these metrics. Sorry, and what we've arrived at now is attempts to do these 1 kilometer distribution predictions that in a way aren't true EBVs yet because they aren't temporal yet, but then you can use other shortcuts, use habitat change, land cover change to make them temporal. And I encourage you to explore some of that on Map of Life or in the Half-Earth Project Map, where this is starting to come together. We are beginning to work with regional and local actors here. The government in Guyana, we just had an MOU signed with the government to support them in their planning of monitoring and national parks throughout the country of Guyana. So this is a great starting point. It's not the end point, it's the starting point to then think about where is the uncertainty, where should we, and how should we sample given the available data and given the remaining uncertainties. But we are developing infrastructure that is helping with that. Thank you. And then dashboards that allow countries in their own planning as to, for example, where are there conservation gaps, where should they not just sample better, but also think about additional protected area engagement. Now, this all stands and falls with the quality of these predictions, and we've worked hard over the last 8 years to develop this module that allows experts to engage around the SDM predictions in particular. There's another great example here by the Humboldt Institute, the Rainer Modellos work. They've been doing that as well and engaging with Colombian experts. Here we're trying to do that at the global scale to iteratively refine these models. I mean, AI and deep learning SDMs are all nice and great, but you got to look at the maps, right? And then realize what's actually going on there. And because otherwise you're also not going to get the traction downstream. You're not just doing wrong and might be actually causing more problems than solutions, right, through your work, but you're also not going to buy-in from the local and regional actors. So we're working a lot on that. We're working on new methods to deal with this group of species for which we just do not have enough data. The best deep learning model is not going to help you directly with these here. What we need is approaches to borrow strength from some of the more data-rich species. And here, graduate student in my group, Shruti Sharma, has been doing very awesome work in using phylogenetic relationships and what we can identify in terms of phylogenetically conserved niches to then make predictions for data poor species from data rich species. And it really makes a huge difference here for these data poor species on the left side and how we're able to improve the models. So happy to chat more with you about that individually. And with that, thank you for your attention.
Okay, we have like 30 seconds for a question. Oh, yeah.
It's loud enough, it's fine.
No, it's for the
Yeah.
Sure. Yeah.
Just to be sure if it is when you make the difference between local and global, in fact you are part of the local with part of the global. You cannot say a mistake, so it's not just local and global, but local and a part of the local who is part of the global.
Don't be confused there, but the point is that to appreciate the full compound significance of a location, of the mountaintops up there, right, which have the Paramount, have amazing endemic species. You got to look at both the local benefits that arise from that, for example, erosion control, right, timber, other things. You all know about nature benefits, right? And the larger global significance that that place has as well, right? The endemic species of which the people here are the sole stewards of the world. And our challenge is to quantify that comparatively across the world, right? And bring that into the conversations. That's the point I'm trying to make. You got to do both and bring them together.
Okay. Our next speaker is Daniel Lopez.
Buenas tardes a todos, yo soy Daniel López del Centro para la Biodiversidad y la Conservación del Museo Americano de Historia Natural. Y hoy voy a presentar nuestro trabajo de expandir el software de Wallace de Comod para calcular los indicadores del cambio en la biodiversidad a partir de modelos de distribución de especies para la planeación y el manejo de la conservación. Este trabajo originalmente lo iba a presentar la doctora Melinda, pero no pudo estar aquí. Entonces les voy a presentar esto, pero como pueden ver, es un trabajo con muchos autores. Muchos de los autores están aquí en este salón o en la conferencia. Es lo que les voy a presentar el día de hoy. Entonces, información actualizada para poder entender temas como el cambio climático o la pérdida de hábitat en la conservación, entre otras aplicaciones. Y pues también sabemos que desde yo se han establecido unas variables esenciales de la biodiversidad para poder armonizar el monitoreo, y con los modelos de distribución de especies se puede estimar la variable de distribución de especies de una mejor manera con los resultados que obtenemos de estos modelos. Estos modelos, como sabemos, es una relación entre las ocurrencias o los registros de ocurrencias de especies con variables ambientales. Estas, a través de métodos y algoritmos, producen unos modelos en el espacio ambiental que luego pueden ser proyectados al espacio geográfico. Entonces, estos modelos también nos ayudan a contestar preguntas que son indispensables en las ciencias de la conservación, preguntas que van desde la identificación de problemas hasta la definición de posibles acciones, pero una de las cosas que afectan la utilización de estos modelos es el grado de incertidumbre que se tiene, porque en cada paso del flujo de trabajo, cuando se genera o se construye un modelo, siempre hay que hacer suposiciones, y estas suposiciones tienen un gran efecto sobre el resultado final. Entonces, esto nos lleva a que hay un gran nivel de incertidumbre con los modelos. Sin embargo, pues existe mucha literatura y hay muchos avances en cuanto a ecología y evolución. en temas de nuevos métodos y algoritmos avanzados, incluyendo métodos de aprendizaje automático, pero es muy difícil para las personas que no están familiarizadas con el código fuente, si no escriben código, no leen código, poder aprovechar estas nuevas técnicas y métodos. Entonces, desde un equipo grande de ecólogos y desarrolladores de software, decidimos sentarse, pensar en esta problemática y crearon Wallace. Wallace es una plataforma de software abierta y colaborativa que permite el desarrollo de modelos de distribución de especies de una forma que disminuye esas barreras que presentan muchas veces trabajar directamente con código, que permite que las personas puedan aplicar en sus investigaciones, en sus esfuerzos de conservación, estos modelos de distribución de especies. También muchos de los autores de Wallace están aquí hoy, en este salón. Y Wallace es una herramienta desarrollada en el lenguaje de programación R con el paquete Shiny, que permite que sea una aplicación web, es muy amigable con el usuario y permite tener acceso a esos métodos y algoritmos de vanguardia de aprendizaje automático para generar estos modelos. Además, como pueden ver, es amigable para el usuario y lleva al usuario a través de todo el flujo de trabajo de un ejercicio de modelamiento, la mayoría de los pasos, guiándolo en cada uno de ellos para seguir las mejores prácticas que se encuentran en literatura y soportadas con referencias. Una cosa importante de Wallace es que es expandible y pueden agregarse nuevos módulos o métodos cuando ya estén disponibles y pues es abierto, gratuito, se puede obtener fácilmente. En este momento existe un equipo que mantiene la aplicación de Wallace, es una herramienta que está activa y tiene muchas opciones que se pueden ir agregando, todo esto de acuerdo a la retroalimentación que tenemos de la comunidad de usuarios. Este es un ejemplo del lanzamiento de la versión 2, donde se incluyeron bastantes funcionalidades nuevas que ya habíamos discutido con la comunidad. Y también, pues, esencialmente lo que les voy a hablar hoy, que es de las funcionalidades que se van a agregar en una versión futura, en una versión que va a ser lanzada próximamente, donde se agregan nuevos módulos que permiten tanto el cálculo de estos indicadores del cambio de la biodiversidad como el uso de productos de sensores remotos para poder refinar los modelos de distribución de especies. Antes de entrar en detalles, solo quería mencionar que este trabajo es un co-desarrollo, se desarrolló en conjunto con la red de observación de la biodiversidad en Colombia, Colombia BOD, que maneja otros sistemas de información de la biodiversidad en el país, pues a nivel nacional y subnacional. Ok, entonces lo que hicimos esencialmente, este trabajo, fue crear dos paquetes de R, los paquetes Mass Range R y Change Range R, que permiten realizar refinamiento del modelo para poder obtener la distribución o la estimación actual de la distribución de una especie y también calcular algunos indicadores. Entonces, el primer componente para Wallace, que está implementado también en Wallace, utiliza este paquete Mass Range R. Y es un paquete que nos proporciona funciones para el enmascaramiento y refinamiento de los modelos ya existentes. Esto a partir de productos de sensores remotos como cobertura de bosques. También se pueden agregar o eliminar áreas dependiendo del conocimiento de expertos o se pueden utilizar otros polígonos o áreas espaciales como áreas protegidas para el mejoramiento del modelo. También tenemos uno llamado indicadores que permite el cálculo de indicadores a partir del modelo de distribución generado en Wallace con otra información adicional que incluye diferentes tipos de indicadores, principalmente pues indicadores como los que proponen la IUCN para la evaluación de riesgo de extinción de especies. Pero tenemos otros también temas de cambio con productos GIS para la deforestación o el sobrelapamiento de áreas, por ejemplo, con áreas protegidas, y esto nos lo permite hacer este paquete. De igual manera, un tercer módulo permite también con datos del modelo de distribución de especie generada obtener métricas multiespecies, que serían como la riqueza de especies, endemismo, e incluso estamos trabajando en el mismo filogenético que no se encuentra ahorita en Wallace, pero sí en el paquete que les acababa de presentar. Otra característica de esta nueva versión que vamos a tener en vivo es que logramos integrar Wallace y biomodelos, que es la herramienta de Colombia Board, que permite construir colaborativamente con expertos modelos de distribución de especies, y es muy importante porque cuenta con registros de observación que han sido revisados por expertos previamente, han sido curados. Entonces, ahorita Wallace permite obtener esos registros ya curados, generar su proceso para obtener modelos de distribución refinados con los nuevos módulos, y esos productos se pueden entregar nuevamente a biomodelos, que se encarga de presentarlos y ponerlos disponibles al público en general. Entonces, pues lo que queremos mostrar también es que a través de todo este trabajo que hemos hecho, se puede permitir o se pueden entregar nuevas herramientas que permitan, por ejemplo, mejorar el proceso que se hace para la evaluación de especies, de extinción de especies, las listas rojas, también para que se puedan hacer evaluaciones de biodiversidad a nivel país o a nivel grupo, o también otras iniciativas que buscan, por ejemplo, revisar el impacto de los proyectos de infraestructura en la biodiversidad, y otros temas de conservación. También quería contarles un poco que en el equipo estamos en este momento en una segunda ronda de financiación con la NASA, en un proyecto donde lo que queremos es integrar estas herramientas del Colombia Board con otras herramientas que se manejan para poder realizar toma de decisiones desde el Sistema Nacional de Áreas Protegidas de Colombia. Entonces, eso es algo que estamos haciendo actualmente, está en proceso, y pues esperamos mostrar los resultados próximamente de esa parte. Un comentario adicional también que quería hacer es que nada de esto hubiera sido posible sin el trabajo colaborativo y el codiseño. Este trabajo, como les conté, tiene muchos actores, muchos participantes, y muchas organizaciones trabajaron para poder llegar a estos resultados. Y el año pasado, en el mes de diciembre, en la revista Bioscience se publicó ese resultado con las lecciones aprendidas y se describe mejor el proceso. Entonces lo pueden consultar si están interesados. Esta mañana también dimos una charla explicando un poco más sobre el proceso de codiseño y colaborativo. Y pues los invito a que descarguen Wallace si no lo conocen. Pueden ingresar a la página y pueden ver toda la documentación que existe sobre el programa. También, si están interesados en esta versión que tiene los nuevos módulos, pues pueden escribir y nosotros podemos compartir las instrucciones, porque todavía no está abierto al público, pero me gustaría que ustedes lo probaran, conocieran sus opiniones. También les cuento que Wallace cuenta con un material para la enseñanza y para entrenamiento. Está disponible la viñeta de Wallace en varios idiomas, está en inglés, está en español también, entonces pueden consultarlo. Este material también está disponible en la revista Species Population del Museo Americano de Historia Natural, donde también pueden encontrar estos ejercicios en formato artículo. Esta revista es abierta y también pueden consultarla sin problema. Tenemos también una versión beta de la interfaz en japonés. Nos gustaría poder llevarlo a más idiomas para que otras personas puedan utilizarlo. Y finalmente, pues quiero agradecer a los equipos que hicieron parte de todo este trabajo. Está el equipo Wallace, está el equipo Biomodelos del Instituto Humboldt, está el equipo del PON Colombia. De igual manera, la financiación de NASA para este proyecto pues, fue muy importante. Y también pues agradecer a todos los socios de Wallace en diferentes partes del mundo, y también a los que han logrado pues, apoyar todo este trabajo. Entonces, pues, muchas gracias. Pueden aquí descargar también Wallace. Si hay preguntas, estamos disponibles, y gracias por el tiempo. para calcular esta utilización del periodo los niveles de equilibrio, simplemente lo activamos en el estos modelos que se generan dependen mucho de los insumos entregados. Si se quieren hacer multitemporales, hay que hacerlo pues, con insumos de diferentes momentos en el tiempo. Pero si tenemos planeado a futuro buscar una forma de hacer modelos multitemporales directamente. Hay un ejercicio del Instituto Humboldt siguió a investigar el que vea de realizar modelos multitemporales, pero por ahora van a ser según el insumo que proporciones. Los datos que tengas, pues puedes hacer también proyecciones a futuro de otros momentos en el tiempo. Si tienes los insumos correspondientes, las capas ambientales, y de igual forma también a otras áreas se puede hacer esa predicción del resultado del modelo que se seleccionó, que van a salir en la herramienta.
For next speaker, one size evaluable. Just a reminder that outside there is the code for presentation.
Good afternoon, everybody. My name is Jose, and I am an informatic researcher from the Institute of Ecology at the University of Chile. And I am here to present biodiversity monitoring strategy for Chile, the origin summer-based environmental reporting and public baseline initiative. I already did this in English, but I've been using Spanish, as I'm sorry. And the main topic of this presentation and our work for the last year has been maintaining the quality and standard of the data generated for the public baseline. So the public baseline is a government initiative that aims to generate accessible environmental and socioeconomic data for regions in Chile. This data aims to inform decision making by governments, the private sector, civil society, and academic research, and support transparency, coordination, and the quality of environmental assessment processes. This initiative began in 2023, and by 2025, there will be the initiative comprised 11 environmental components listed here: human environment, protected areas, landscape values, physical environment, activities, and environmental impact, cultural heritage, land use, and tourist value. And the one I left behind are the biodiversity one, marine ecosystem, terrestrial ecosystem, and continental aquatic ecosystem. The third tender to collect these environment components is Antofagasta, which the University of Antofagasta is organizing in collaboration with the Institute of Ecology. The work group of this project are the 11 components listed in the slide, and the geomatic team in charge of the data quality of the information derived within the data standardization process, systematic and easy, we develop a platform for researchers to develop the data. This allows to generate a structured database, like progress and analyze missing zones. This also allows for the disparate adaptation of all this information to the minister. Here we show how each environmental component work will be flexible. Here, leaders of the different task groups can upload the data, which is checked, structured, and integrated. Once the file is uploaded, they must associate metadata, keeping the integration and practice information from further analysis. Here's a diagram of the structure followed for the process of the biodiversity data of this project. Raw data uploaded to the platform. The study is accepted, the manager researcher has associated the data with the form. The event occurs in taxon data in the right panel. Follow the Darwin Core standard, in particular, presenting events are structured in the Humboldt Core standard. proposed protocol for continuing to monitor biodiversity sampling areas. These are intensive monitoring programs. There are three main components. The campaigns, which define the group monitor, replica stations. Stations have a designated area and can contain multiple replicas. Replicas must use the same methodology of monitoring type. The form includes information of the dimensions and location of the replica station, and tier 1 is the currents, which are the specimen at a particular time of place. The environmental superintendents estimate for the name in Spanish as a standardized protocol in the form 343. This is required for all environmental assessment in Chile. Actually, in Chile, there are more than 15,000 of them. This is an unprecedented amount of sampling and data, which we present as the basis for the biodiversity monitoring system. We intend to take advantage of all resources to start biodiversity monitoring study. Sampling events in the context of species awards data are at this level at the small and large area inventory. This means that these protocols are asymptomatic or the absence of the same way sampling events are informative of the abundance and distribution of the species. Our challenge is to bring our view or raw data into the context of essential biodiversity variable. In this way, we will support our rating indicators of trend biodiversity changes, which is a global important but more critical for us is national link in the context of the commitment. And in summary, what we're doing is to integrate the SMA 343 format, which is to record and collect sampling events into a context of data integration, which is provided by the Public Baseline Initiative, to be able to offer national tools for reporting progress and global goals and targets. We are going to do this by standardizing this form, making the mapping to the common core extension. And these are the some images. Thank you very much.
We can wait like a few minutes for start video. So just keep the time. Yeah.
Hey everyone, my name is Beth Gerstner and I'm a biodiversity scientist.
So the question for what about how to think about it first. How driven has this research been by interest from governments, and are you sort of designing the work to feed into government planning?
Hey everyone.
That's not the government is funding the program, and we as experts for it are the ones that have to decide which areas are going to be. There are a lot of work that I am not the one that do it. As a team, I am the informatics researcher, so I'm using the background and things like that. But my colleagues are the ones that decide. They do inspections and executions. If anyone knows who is how, they are the ones that can accept it. But the whole project is absolutely a government subject. That's
So our next presentation is a review, so we are so
Okay, guys. Sorry. So the next speaker is Beth Kirchner. She's doing like a video presentation. So
My name is Beth Gerstner, and I'm a biodiversity scientist at the Center for Biodiversity and Global Change at Yale University. And today I'll be talking about how we're completing 1 kilometer global species distribution EBV for all taxa, progress and challenges. So biodiversity encompasses all life on this planet, but in many places it's rapidly declining due to human threats like habitat loss and climate change. And without intervention, we'll continue to lose more. So to bend the curve of biodiversity loss, we first need to increase conservation efforts. And this was recognized during the UN Biodiversity Conference in 2022, where they set a goal to protect 30% of the planet's land and oceans by 2030. However, this raises a critical question: which 30% do we protect? To address this, we first need to understand the spatial distribution of biodiversity. Not all areas contribute equally to conservation, and simply designating land or oceans as protected does not guarantee species survival, especially under future climate change. That's why we need high-resolution species distribution models that not only map current biodiversity patterns, but also forecast changes under different climate scenarios. To make conservation decisions, we need models projections over time, and relevant scales. Essential biodiversity variables, or EBVs, provide a global framework for tracking biodiversity change, and high-resolution species distribution models can be key to generating them. By mapping species through space and time, SDMs underpin indicators like extinction risk, protection, coverage, and ecosystem services, which are essential for guiding conservation, tracking global targets, and informing policy. But as you can imagine, modeling the spatial distribution of all biodiversity across taxa, regions, data qualities, etc., is a massive undertaking. And at Map Life, we built pipelines to generate high-resolution SDMs for thousands of species at a time. But modeling at this scale brings a host of challenges, such as biased and uneven data, model evaluation issues, and species-specific knowledge gaps, just to name a few. And that's why we need more than just automation. We also need collaboration. So today I'll walk you through how we're tackling these challenges and how we're bringing in expert knowledge at scale through a system we've developed called Expert Tools. And this is a structured platform for reviewing and improving SDM predictions with input from those who know the species and regions best. Here at Map of Life, we bring together data from different sources to understand species distributions and calculate biodiversity indicators. But what you see here is just a subset of the data that we rely on, ranging from global occurrence databases to exoterm range maps to trait and environmental data sets. And integrating these sources is what really allows us to model biodiversity comprehensively across taxa and regions. In this vein, one of the center's core initiatives is to develop extremely high-resolution global SDMs that integrate multiple data sources for terrestrial vertebrates, select invertebrates and plants, and marine species. To model at the scale efficiently, we have built a semi-autonomous workflow that streamlines the entire process, from taxonomic harmonization and data cleaning to model development and performance evaluation. Once we configure the modeling parameters for a given taxonomic region, the modular pipeline runs autonomously. This approach enables us to generate models for thousands of species rapidly and consistently while making it easier to troubleshoot specific taxon issues. These models support a wide range of applications, from calculating biodiversity indicators to informing spatial conservation priorities. However, modeling at scale presents significant quality control challenges. While statistical performance is relatively easy to assess, ensuring that models accurately capture spatial patterns and species ecological realities is far more complex. To address this, we incorporate a human-in-the-loop expert review process that supplements quantitative evaluation metrics. Models undergo expert validation to assess biological realism, identify spatial inaccuracies, and guide targeted refinements of models. By integrating expert feedback through the structured review process, we can improve model accuracy and ensure that predictions remain actionable for conservation applications. Importantly, the workflow is designed to be iterative. We can easily adjust model settings, retrain models, and rerun entire taxonomic or regional pipelines as new data, methods, and expert feedback become available. This means our modeling framework can function as a living system, one that can be continuously updated and improved over time, rather than a one-off modeling effort. One of the biggest challenges in modeling species distributions at scale is the sheer computational demands and the complex multi-step nature of the workflow. As I mentioned before, we have a semi-automated modeling pipeline allowing us to efficiently deploy models for thousands of species across broad extents. And by caching all of our intermediate steps, we can easily analyze outputs, troubleshoot modeling issues, and refine our predictions. Additionally, running our workflow on both high-performance computing systems and the Microsoft Azure cloud has significantly enhanced efficiency and scalability. After cleaning and preparing the data, we run presence-only SDM similar to Maxent, but with a key improvement. We use expert range maps as spatial offsets. This anchors the predictions in expert knowledge and improves ecological realism, especially for data-poor species with sparse or biased records. Offsets give the model a baseline of where species are expected to occur, reducing overfitting and improving predictions even with limited data. To ensure all species are represented, we are also working to combine multiple data sources, including expert maps, phylogenetic and functional trait data, leveraging well-documented species to fill gaps for poorly known ones. Even with expert input, model quality depends on data quality. Presence-only data often reflect where people survey, not necessarily where species truly occur. To correct this, we use target group background sampling, which pulls records from species with similar traits, thinking about nocturnal or arboreal birds or taxonomically related species, to generate a kernel density bias surface. And this ensures background points approximate survey effort, reducing overfitting and improving ecological realism. While TGB is well established, we've now applied it globally for birds, which is a first, enhancing model calibration and paving the way for expansion to other taxons. So now let's step back and look at our modeling progress thus far. We've completed initial models for North, Central, and South America and Southeast Asia across amphibians, reptiles, birds, mammals, ants, woodweeds, butterflies, and trees. And we've also modeled Amazon basin freshwater fish and expanded bird models in Africa, Australia, and Eurasia. Altogether, this covers about 28,203 species, with plans to expand to plants, more fish groups, and marine species. Overall, model performance is strong with average AUCs of 0.85 to 0.91, and 82% of models having AUCs greater than or equal to 0.80. We're continually refining workflows to boost robustness. While AUC gives a quick sense of quantitative performance, spatial performance is a lot harder to assess. And one key way we evaluate this is by examining spatial patterns of species richness and rarity across different regions. After model runs, we first examine species richness and rarity to assess broad scale performance, focusing on mammals where coverage is most complete. We modeled 1,454 species across the Americas. Richness peaks in Colombia, Ecuador, and Central America as expected. We also mapped rarity, highlighting areas rich in range-restricted species. And while these patterns provide powerful insights, we know the models aren't exactly perfect, especially at the scale that we're working. So next, I'll walk you through the steps we take to make them more robust and methodologically realistic. While statistical evaluation is straightforward, spatial validation is far more challenging, especially at scale. To address this, we're engaging experts through our expert tools platform. Without species-specific training, global models can produce errors like overprediction or overfitting, which metrics alone might miss. Models is a great example of the importance of expert validation at the national scale. Building on this spirit, expert tools aims to provide a standardized, scalable framework for integrating expert input globally. We've completed this process for North America and are now expanding to other regions. These are some of our existing and growing partnerships, most directly using our SDMs. In particular, we're now focusing and working with the ASEAN Center for Biodiversity to begin using expert tools, enabling regional experts to directly review and refine models. And we'll be doing something similar with the Guyanese government. So a little bit about our expert tools platform, designed to bring expert knowledge directly into the modeling workflow. Once models are completed, they're uploaded to the expert tools platform, which is fully integrated into the MapReflect website. Right now, our primary focus is on collecting and organizing expert feedback, but the next step is to automate responses to certain types of input. For example, having flagged issues like false absences and overfitting automatically trigger model adjustments. And I'll walk you through an example of how that works. Once a species is assigned to an expert, they can begin reviewing the model and submitting feedback through the platform. Experts can flag false presences, false absences, irregular range boundaries, things like that. Here's a quick example of how this works in practice. This is an initial SDM we produced for the Olinquito, a cloud forest endemic in Colombia and Ecuador, and a species listed as near threatened by the IUCN due to habitat loss and climate change. As an Olinquito expert, I know there are confirmed occurrences in the eastern Cordillera of Ecuador, but the model doesn't show them. That's a false absence. In expert tools, I can select false absence and draw a polygon around the area where the model is under predicting. In future iterations, we plan to automate how this feedback is used. For example, once a polygon is submitted, the system can check for known occurrences within that area. If those points were accidentally excluded, say during spatial thinning or filtering, the model can be automatically rerun, retaining and prioritizing those occurrences. And this will allow us to respond to expert feedback quickly and consistently, and scale the review process across thousands of species. So once we receive that kind of feedback, flagging false absences, for example, there are actually a number of things we can do to improve the model. The workflow is flexible, and we've built in multiple levers that we can toggle depending on the species and the kind of issue that's been identified. These are just a few examples. For example, if we have erroneous occurrence records, we can remove those records. If there are artificial shapes and lines, we can alter offset parameters. If the range map seems to be incorrect, we can add an alternate, turn off the modeling offsets, so on and so forth. So going back to that Olinquito example, if the model fails to predict into a region like the Eastern Cordillera, even though we know species occur there, that's a problem. It means that part of the species range, such as this, could be excluded from conservation planning, monitoring, or resource allocation. For rare or range-restricted species, even small omissions or modeling errors can significantly shift conservation priorities. So this kind of expert modeler feedback loop is essential for getting our instruments right and having confidence in our downstream products. So as we look ahead, we have 4 main goals. First, we want to expand coverage, modeling more taxa across Africa, Eurasia, and Australia to fill our current gaps. Second, we're building a system for iterative modeling, quickly integrating expert feedback so models keep improving as data and methods come in. And this will be an autonomous workflow that gets triggered on the fly. Third, we'll add temporal dynamics, updating models through time to move from static maps towards projections and forecasting. And finally, we'll link to EBVs and indicators, generating standardized policy-relevant products to support global biodiversity monitoring. Together, these steps will help build a global modeling framework that's both rigorous and actionable for conservation. And with that, I'd like to thank all of our funding agencies for supporting this work that we're doing here at Yale Center for Biodiversity and Global Change. And now I'm happy to take any questions.
You have a question in presentation, whether it's
Or is Steph online? If she's online, how does this work? Sure. No? Okay.
And she comes up.
I'm sure the details in the paper, but when you say well sampled cells, when you draw on well sampled cells, to inform distributions of species or species that are well sampled.
You mean the borrowing strength example that
Yes,
you brought up
yes.
earlier? Huh?
You had some said something about well sampled.
Yeah.
What is well sampled?
Okay, there's actually no threshold applied. It's literally you're grabbing a portion of the phylogenetic tree, and usually we take like 30 to 60 species chunks, and you're looking at how strongly is the environmental niche conserved. That's simplified things a little bit here. So how similar, how much is proximity on the phylogenetic tree associated with proximity in environment space, as indicated by the data you have, right? And that will be mostly driven by the species results of data. You can then you then know which within that tree section, which species are expected to be similar in niche to another. And then that includes even species for which you didn't have data because they're part of another larger clade, right? That you can then use to make predictions for those undersampled species. That's the idea. It's incredibly powerful. There's a whole literature on environmental conservatism, right? We'll debate about it as well. So it's not so easy. We're simplifying it a little bit here, but in terms of a pragmatic way forward, there is no doubt, right? If you hear there are two hummingbirds that are occupying a sister species, one is on this side of the Andes, the other one on that side of the Andes, right? They'll occupy, they look separate at some point, that's why separate species, but you see in that clade tends to evolve very slowly in terms of the environmental associations. And so if one of those species is really data poor, there is at least something you might be able to infer about it, right? Not to perfection, but at least something to not leave it behind. I think one of the biggest problems, actually, much of the species distribution model work, including some of ours in the first steps, right, is that we just tend to leave out. We talk, well, we got 10,000 species, right? But what if your actual scope is 15,000 species, right? And where are those 5,000 species? They tend to be in the tropics, they tend to be the small range species, they tend to be the ones far away from roads. And if that's what you're pushing, that's not an EBV. Because you're missing species in a non-random way, and any metric interpretation, any planning that happens from there on would be biased and be leaving out some of the most critical areas, right? That tend to be the ones with data poor species that are narrow ranged and or data poor for that reason alone or far away, etc. So it's really, 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 really important. So that's what we're spending a lot of our time on to try and address.
Yeah, maybe on this point,
Sure.
yeah, just a follow-up question, really. I would have thought traits would have more predictive power than phylogeny. I was wondering what you see as the phylogeny is more available?
Exactly, right? Because once you get to traits, again, you tend to have trait gaps in those groups that are undersampled. And I mean, yes, for birds, we could run with a whole suite of traits or mammals as well, right? We've done some of those initial data compilations and played with that. But then you also got to figure out how traits are related to niches, and we are exploring that as well. And the method actually would work with traits as well. But we tend to get much more robust completeness actually increasingly with phylogeny. So for vertebrates, we've put together this first whole set of first complete extant species sampled trees, right? Phylogeny frameworks, in order to be able to do this. So that we started that, like, this was all like step by step by step, right? You got to build up the knowledge base. And obviously now genetic data is exploding. We're getting this increasingly for other species groups, near completeness, and then you can again use methods to place some of the missing species. I'm not saying this is I think it's a little silver bullet, but it's not the solution, right? But it's one way to just bring them in and most importantly, not leave them out, right? And be able to say, well, they're in, and yes, they have larger uncertainty still. Obviously, your undersampled species will have a larger uncertainty still, but at least we're able to bring them in somewhat, not leave them behind.
I think we don't have time for a question.
Yeah.
Thank you. So, write some questions for 5 minutes. Based on the abstract. At first, we thought that maybe came here to talk about it, but now we think it's better that if anyone in the audience wants to join the discussion, so you are welcome to do it. So one of the questions that I think we could talk about, how can interoperability among platforms such as Biomodelos, Map of Life, and Eagles be improved without losing traceability or data quality? Should there be regionally agreed minimum standards for the data and taxonomic processing
From the position of ignorance, that's, I think, sort of a value-based response. So, yes, absolutely. Second question, part of the question, absolutely. There should be working towards standards. I think the government data package, is in the new development. In terms of the integration of these different platforms, I have no idea, but I think it's a balance between very appropriate and very necessary local to regional to national scale development of particular platforms that are prioritize country priorities and differ and that develop local capacity. And there might be a stage at which it's useful to integrate them, but there might be a stage at which it's perhaps too early to integrate. I think there's benefits both to having them independently. Perhaps, I don't know, my gut response is that integrating them too early might not be the right idea.
Yeah, happy to comment on that as well. I think they're all really interesting examples that have some overlapping, some commonalities and opportunities to double down on those together, but then also some really distinct attributes. So eBird, for example, is able to really push the boundaries on some things that others just cannot, right? Because with birds, you have such outstanding data and protocol, the way it gets sampled, right? It's actually one of the few data types that you can actually sort of absence information through the protocol that they developed. And you can try and push it all the way to the abundance side, right? Or attempt to do so. We can put a little bit about this. So there's certain, in terms of supporting maximum innovation, right? eBird can focus on some things that others just cannot, and push the boundaries and move us along, right? The community along. If you go like opening eyes or showing things, or some discussion. Identify models by focusing on, and I'm just shooting from the hip here. That's okay, but biomodels was one of the first, certainly tropical, but maybe even global efforts to try at the national scales pull everything together, right, and do the end-to-end from raw data to models to experts in an operational level, and do that with a clear focus on the country and the local experts there. I think there's certainly opportunity and ultimately need to work more closely together when it comes to certain common products, right? I see that, for example, expert feedback, and that applies to other efforts as well, right? So it's a real, that's really the very good. That one expert, right, that really knows that species or that species group, or is in Colombia or in Bolivia or in Nigeria, and just knows that corner and its species. And it's extremely inefficient and actually a disservice by our community and the respective efforts to then for different efforts to approach that same person for their input, and that data to that input to then disappear in different silos and not being shared, right? Or the quality control around the data points. How many GBIF data points have been multiple times assessed and thrown out and determined as problematic? And that information never made it back to GBIF or to the original providers. It's sitting somewhere in silos. All of these efforts do their respective quality control, and never does this really come together. So that's what I'm concerned about. That there's a lot of duplication, a lot of inefficiency, and we need to, especially as the modeling gets computationally easier and almost like a commodity as you think about some of these approaches. That's where we need to focus, and we need to pull together there in terms of our platforms, so they don't have to be one and the same platform with the APIs, just connect them up with one another, so that any of this feedback and improvement of data quality gets shared and ultimately lifts up the larger birds. Doesn't mean it all needs to be one product or one place. Anyway, those are just my 2 cents.
Anyone wants to give an opinion about it?
Just a quick comment. I'm not sure if it is a question or not, because I understand the difference on those platforms and the need of having those differences because they have different alliances. We communicate with what we need, but at the same time, for users, for decision takers, it could be really difficult sometimes to decide with so many platforms that we are seeing in this conference, not only in this moment, in this part of the conference, but along these days. There are so many that it could be also very difficult sometimes to decide, okay, which one should be used? Which one is going to have the best data for the decision taker or for the community that is going to make an effort to enter, to understand, to get into. So on how to use the basic data to develop analysis and knowledge on that data. Not so much on what the data is, but more what the information is being gathered from that data could be maybe more useful for decision takers and not like separate all platforms on the data, but on the information. I don't know if you, that's a little bit what I understand also what you're saying, but it's kind of a challenge for using the data with so many distributions.
No, I totally agree with you that for a user it's confusing. Like, but I do see that they are very different, so I think it's an issue of communicating. You know, if you need to assess something global, you know, maybe map could be good if you want to develop by yourself the models. Why not? If you want, you know, to go beyond with the good models, either, but it all depends on what. But I think that is a responsibility also of the people that are putting that information out to explain like the use of that data, and that is something that the information. And I think that is something that we also have to have in promoting the model about in the atlas books. It's like, you know, what is this information for? What is telling you? What is not telling you? How can you use it? I think that is something that we need to work. But I agree that it's difficult to put them together, and because they have also been evolving in different ways. Some of them close by, like Guanas and the modelos that are like now, you know, good integration, but then there are others that are more difficult. But some charming topics are there for all us to work together as a community, and I think that is the value of the population EBV working group within GEO1. And I think it also needs, you know, we're talking with Melody, how to really get back again in a very active way that group of working and be again like and users to be part of that group would be fantastic. That would really help us decide where to go, where to work, how to work.
And actually, maybe actually develop a bit of a typology or an overview, right, of the different efforts out there. Could be one of the, that could be one of the things that the GeoBond working group on this topic should tackle. Yeah. So we're happy to hear from you to grab us, Melody, myself, Maria, or anybody else. But if you want to, we've been trying to keep that ship going, right, within a GeoBond, but obviously we volunteer our time if we get distracted. And so any suggestions in that regard, but that's something you have popping up. It's a real potential.
Okay, thank you very much. Well,
No, I have like just one last comment. I mean, talking about one possibility of the integration of, for example, quality of data. Something that, for example, what it does and does is, for example, experts who get the data, they remove records that they think they're incorrect for some reason. But for example, most of the data is not reported to be. So it's something that I think is important that probably these two platforms and other platforms can look into the way, for example, the work of the experts can contribute not just for the models in Colombia or global, if they are using those bad quality records for other models and other issues.
Yeah. Okay, thanks to everyone stressing a little bit what you just mentioned. I think that definitely we need to kind of like speak the same language, because otherwise in the different platforms or a bit contributing to definitely just say, if not as user, as particularly marine modeler Antarctic user, then it's really hard, because when there is not so much available data, you need to definitely try to do what it is with the things that are available. So then, when you're combining different things, it's very hard, because when the topic is not the same, or the way how samples are being done, or the way how the resolution is being done, then working into a specific region, then it's harder. So I think that definitely for the main question that you were starting with also the discussion of uncertainties, or which is the tax that we need to put an eye on, I think that in the end we need to think globally, but also like being able to deep into regions that maybe are not the most famous one, or maybe they are, but we are really far away of getting data. So I stress once again this, like we need to standardize the information and to make it also global, and to make it, as you say, Maria, also communicating the differences in order for us as users to be able to just from. Thank you for allowing me.
Thank you. And I just wanted to say, if you're not a member of the GeoBond Species Population Working Group, it's entirely open, and you can just go onto the GeoBond website to the Species Population. There's a membership page, and you can join the working group, which means you go onto the mailing list, and then you'll receive mails. It's a good way to stay in touch. But otherwise, thank you very much, and thanks to all of you for participating.
Yes, no.